Welcome to Brain Ponderings, your window into the world of brain research with unfiltered conversations with scientists at the forefront of neuroscience, neurology, psychology. I'm Mark Masson, the host. Today, my guest is Rajiv Ratan. He's a professor of neurology and neuroscience at Weill Cornell Medicine and CEO of the Burke Neurological Institute up in New York. And his own personal research laboratory focuses on cell stress, responses to cell stress, adaptive responses in the context of trying to figure out ways to reduce damage, for example, following a stroke or traumatic brain injury, and importantly, to enhance recovery. And maybe some of the things he's working on now will be translated to humans hopefully pretty soon. So welcome, Raj.
Yeah, thanks for having me, Mark. It's so nice. It's a real privilege and honor for me to have this conversation with you because I've admired your work so much for so many years um, and have been heavily influenced by it.
Let's start with your background. So your parents they immigrated here from India, is that right?
Yeah. My dad came in 1955 to train as a heart surgeon in Pittsburgh. And he went back in 1958 uh, and he knew my mom for 2 days. They got married. They came back and
A
and they
typical Indian.
that's right, arranged marriage, and an arranged marriage that lasted sometimes very blissfully and other times not so blissfully for 60 years. And they loved Pittsburgh. It was a great, really welcoming place for immigrants. The year I was born, which was 1960, and that was the year that the Pirates beat the Yankees to win the World Series.
Was that Roberto Clemente? Was he on?
Well, he was, but later. So the story was that Bill Mazeroski. So the game was tied going to the bottom of the ninth inning, seventh game,
Oh.
and Mazeroski hit a home run to win the series for the Pirates. So they, I think it presaged a lot of my life because um, I I think for them sports became an immediate way to assimilate. And as sort of a child of immigrants growing up in America, I was an avid athlete. I was always participating in different sports, and I think it was a great way to fit in.
So they had your parents had to learn the rules of baseball and football rather than cricket.
That's right. And it's amazing. Some of my fondest memories were actually watching, sitting on the floor. My dad was on the couch. I mean, he was a heart surgeon, so sometimes when he was home, he was pretty tired, but watching football games with him. And so that was an other fringe benefit for me of being sort of an avid sports participant and sports enthusiast. And I'm glad to say that that's something I've passed on to my kids.
Now, did your parents? Well, it's your dad, surgeon. So I guess they encouraged you to go to medical school or not?
um, well, I think you know this story pretty well. Is that um, I was I was told I could do pretty much whatever I wanted, but really the reality is I was either going to be an engineer, a doctor, or a lawyer. You know that the socialization is very strong, and I think I was obviously much more driven towards math and science, and I was very fortunate. I ended up going to Amherst College as an undergraduate, and they were the first college in the country to actually have a neuroscience major, and a guy named Steve George. And it really paradoxical because you think of liberal arts colleges, as doing everything that's kind of not practical and differentiated. And yet I had a great opportunity to become a neuroscientist as an undergraduate. And this was, I started in 1977 and I graduated in 81. So as you know, there weren't a lot of neuroscience programs, at least at the undergraduate level
Right.
at that time.
Yeah.
So it was really a treat. And so I think I went through the neuroscience program. It happened that actually the neuroscience course is almost perfectly overlaid with the courses to go to medical school. But I had a chance to do a senior thesis, and my thesis was focused on the role of cerebellar stimulation in psychiatric disorders. There was a guy named Heath at Tulane who'd been doing this for patients with schizophrenia. And so my thesis was ironically using 2-deoxy, radio-labeled 2-deoxyglucose to actually look at what the impact of cerebellar stimulation in rats is on the cerebrum. And in writing my thesis, I had to learn all about the cerebellar circuitry. And
Oh,
so I read a lot of papers by a guy named Rodolfo Linas.
yeah. Yeah. Yeah.
And
And
so
we'll talk about calcium at some point in that conversation.
yeah, well, but the interesting thing was is that one of the reasons I went to NYU was that I wanted to work with Rodolfo, and I
Well,
went.
he had kind of a reputation, that being kind of.
That's right. So I went into his office, and he said to me, he said, "Are you really interested in understanding the nervous system, or do you think you're just going to come and play around and do internal medicine?" And I said, I don't know. And he said, well, you can go down the hall. There's a guy named Mike Shalansky who might want to have you work with him. And so I ended up fortuitously working with Mike Shalansky. And that's where I started to study calcium.
And that's where our common interest first
Yeah,
started.
that's right.
Because we both, at that time, so Roger Chen had just developed these fluorescent probes based on EGTA, this calcium chelating molecule, but he modified them so they fluoresce when
Right.
calcium binds, or the fluorescence changes anyway. And then the other important thing, though, for the initial studies, like you and I both started with cultured cells, right, is he put I think a methyl group on it and made it lipid soluble. So
Right. Right. So they were all the as you remember, the progenitor was Quin2, which was essentially EGTA, and which had acetoxymethyl esters that derivatized all the carboxyl groups. And so it would diffuse into cells. Non-specific esterases would cleave off these acetoxymethyl esters and expose the carboxyl charged groups, trapping the dye in the cell.
yeah.
And one of the reasons why I think Michael was so interested in, and another person who was very influential as I was studying calcium was Fred Maxfield. And Michael was really interested in the cytoskeleton and neurons. But and the question was, can calcium be spatially localized in cells in a way that allows vectorial disassembly of microtubules? Because one of Michael's claims to fame was to purify tubulin. And so the problem with Quin2 is it had very low quantum efficiency. And so you needed a lot of dye in the cell, and it tended to diffuse gradients. And in order to visualize gradients, we ended up studying what was the aplesia of mitosis, the endosperm from the African blood lily. And there are beautiful pictures of this, I think, in the halls of the NIH. But what you can literally see is you can see if you take these endosperm, you can see the chromosomes moving. That's how big they are with the naked eye. And so we were able to load Quin2 into these cells and show that during anaphase, when the chromosomes were separating, that there are areas of increased calcium at each of the poles that is driving the disassembly of microtubules towards and the chromosomes movement towards the poles, but allowing assembly in the interphase zone so that it could push things apart. And I was actually the second author because Charlie Keith started that work, and it was published in Nature in 1985. And then as Fura was developed, we moved on to showing similar observations in eukaryotic cells. And but I learned so much about. I mean, it was an incredible department with fantastic graduate students who were with me, and I learned so much about technique development, about assays, about how when you build a unique piece of equipment like this, it was essentially running 24 hours a day. So there were times where we would do experiments all during the night, and it was really fantastic scientific training. It really only fueled really a love of being in the lab and experimentation.
And that
So
was, you didn't mention, that was at NYU.
that was at NYU, yeah.
And,
And yeah,
and then for your, you were in Baltimore for a while, right?
yeah, so I was in Baltimore for 7 years, and I was fortunate, very fortunate, to do my residency at Hopkins, and I think you know that group extremely well. And Hopkins is it was an incredible place to train as a physician. And I think the institutional spirit and just I think a place where great science segues so seamlessly into compassionate care, I think is so rare. And I really loved being in that environment and training in that environment with Dick Johnson and Guy McCann and Jack Griffin, and it was terrific. And then you'll uh, then when I was getting ready to actually go back to the lab, I since I was interested in calcium, and I called Saul up,
Yeah.
and it wasn't a very coherent um, pitch that I made. And Saul, he's extremely perceptive, so he said, "I don't really have any room in my lab, but I have, but one of the people who worked with me has room," and it was Jay Baraban.
Yeah.
And it turned out to be a real stroke of luck, because at that time, one of Jay's postdocs was a guy named Tim Murphy.
Yeah.
And Tim and initially I was going to study calcium, but I think in talking to Michael and Fred and a lot of people, they said, everybody's studying calcium and cell death. You should really try to find something else. And so I got to Jay's lab, and Tim was there. And as you probably remember, while Tim was with Joe Coyle, he elucidated this novel model of glutamate cytotoxicity, which was non-receptor mediated, and that was dependent on the Xe- transporter. And these are really the seminal studies that laid the foundation for what we now know as ferroptosis.
And so that got you interested in glutamate, in a kind of a unusual way, got you interested in oxidative stress.
Right. Well, it was, you're right. It was so, when I was looking around at what I was going to do, it was so obvious to me that the problem with the field of the problem with understanding oxidative stress was the lack of appropriate in vitro models And if you looked at it, and I think you probably remember this as well, if people wanted to try to model oxidative stress, what they would do is they would add non-physiologic concentrations of oxidants to cells.
Hydrogen peroxide and
Yeah, and nitric oxide and whatever else, the hypochlorous acid, whatever else they could find. And what was great about this model is that because glutamate blocked the uptake of cysteine, the oxidized form of cysteine, it ultimately depleted the versatile antioxidant glutathione,
yeah,
generated an imbalance between oxidants and antioxidants, and really just used the endogenous oxidants to drive death.
yeah.
And so it was, Tim said to me, I'm much more interested in synaptic plasticity. I don't think I want to study this model anymore. Why don't you here have at it? And while I was a resident, I was very fortunate to make the observation that cell death in this model could be blocked by macromolecular synthesis. And that suggested that oxidative death could be programmed.
And that, so you put in inhibitors of protein synthesis,
Right, that's
like
right.
cyclohexamide or something.
Cyclohexamide, actinomycin
Yeah.
D, and we were able to demonstrate that they would protect neurons. And as you know, it was a little bit of a paradigm shift because, I think and it happened at a time where a larger paradigm shift was happening, and that people were becoming interested in apoptosis.
Yeah.
And Korsmeyer had identified BCL2, and it was apparent that cancer was the result of a lack of death, and it was obvious to, clearly obvious to you, and maybe many in the neuroscience field, that the flip side could be apply. And I think what was really fascinating is that, and I think that's what catalyzed an interest in transcription. But interestingly, we showed that the effects of macromolecular synthesis inhibitors could also be ascribed to simply shunting cysteine from protein synthesis to glutathione. And I think that we initially we initially had one big paper. We sent it to Neuron. I think the people who were strongly in favor of the idea of thanogenes, like Gene Johnson, didn't like that idea that maybe there were other metabolic pathways. So it got rejected, and we separated it into two papers. One went to the Journal of Neuroscience, and the other went to the Journal of Neurochemistry. And I think it's sort of a great example of how something topical can be published. Doesn't have to be published in Nature and Science for it to sort of get a lot of attention, because that paper was very highly cited, and I think helped me get my first job much more quickly than I otherwise would have.
So, I think in previous podcasts at some point we've talked about mTOR pathway.
Yeah.
So is if you inhibit mTOR with rapamycin, does it have the same effect as inhibiting protein synthesis with cyclohexamide?
It does.
Yeah.
So it's protective as well. And activating ER stress has the same, and all the activators of the integrated stress response. And the evolution to that idea actually is an interesting story that I may have told you about it. So, after I think you and I both were very interested in NF-kappa B.
Yeah.
And I think that you were finding that it was actually neuroprotective, and I was finding, at least in a virus-induced death model, that was actually propelling death. And so we got very interested in the idea, because it formally still was possible that because macromolecular synthesis inhibitors were so protective, we were really interested in what was the ROS messenger that was unleashed when you depleted glutathione that could activate transcription factors like kappa B or Jun that ultimately might lead to death. So we did a kind of simple-minded and naive experiment. We took catalase that we bought from Sigma, and we loaded up the extracellular bathing medium with catalase. And the idea, as you know, is that peroxide is uncharged, so it diffuses through membranes. So the thought was, is if you load up the medium with catalase, that you would create a sink and protect the cells. And what we found is that catalase from Sigma could protect everything under the sun. It was protective against serum deprivation, staurosporin, everything. But if you look at that preparation, if you were to put it in solution and look at it in a 15 millimeter conical tube, you would see that preparation was really dirty. So what we did is we did a experiment where we inhibited catalase activity, and the protective effect didn't go away. So the first thing I did is I went over to see Rick Huganier, and we started, he had a number of columns that they used to purify peptides. And so he said, sure, you can use it. And so we started a purification on our own. And sure enough, we were able to show that if you get rid of the catalase, the protective effect is there. We put it through a size exclusion chromatography, and we were starting to get to a purified activity. And then I was fortunate. So Lee Rubin.
What was the source? So you bought this from Sigma, that chemical company. And
Yeah.
what was their source? I mean,
liver. It was liver.
Liver.
Yeah.
So there's something else from the liver probably that's protective.
That's right. So we ultimately were able to purify so we did isoelectric focusing followed by gel electrophoresis. And we identified a 36 kilodalton protein that was present in 3 non-sequential bioactive fractions. It wasn't protective in any other fractions. We cut it out. This was with Fred Esch and Lee Rubin at AZI at the time, and we sent it to Bill Land at the Harvard undergrad. And it's 202 amino acids a sequence. It's all identical to arginase. And it turned out that what arginase was doing, so arginase is a metalloenzyme too, so it probably co-purified with catalase using whatever Sigma used. But we found
does arginase, it doesn't have selenium in it, does it?
No.
So
we initially thought, oh, it's arginase degrading arginine. Maybe that means that NO is the relevant. But as you probably know, as you probably know, at that stage of development, the cortical cultures we had didn't express NOS. So and we were able to demonstrate that if you took any amino acid degrading enzyme, you could actually protect the neurons. And ultimately, we showed that what the amino acid degrading enzymes were doing was inhibiting depriving amino acids, activating a kinase called GCN2, which is an upstream, one of the part of the integrated stress response that then phosphorylates EIF2 alpha, inhibits global translation, and that then protected. And we were able to correlate the inhibition of protein synthesis perfectly with the protection by arginase. And I remember giving a talk at a symposium in North Carolina, and Gene was there, and he came up to me afterwards, and he was one of my scientific heroes. He said, Raj, that was a real blunderbuss. you spent 3 years purifying that contaminate, and all you did was find another protein synthesis inhibitor. And to some extent, he was correct, but I think it got us interested not only in the protein synthesis inhibition part of it, but so as you probably know, with the integrated stress response, you get the paradoxical translation of this transcription factor ATF4.
Yeah.
And so it actually got us interested in ATF4 transcription, and that's another long story.
And so the general idea is by imposing an external stress on the cells, you can activate the intrinsic mechanisms which cells evolved to protect themselves against a range of kinds of stressors.
Yeah, and yeah, exactly. So actually, the formative experiments that we did that really got me interested in this idea, I wish I were smart enough to have thought that's the direction I should take the lab. But the story is really, it's another one with twists and turns that will surprise you. So one of the first questions that I asked when I got into the lab full time, it was to take this ferroptosis model and actually
And Raj, can you describe, so ferric or ferrous refers to iron, and ptosis refers to falling off or dying, right? So
yes,
who coined the term?
it was coined by Brent Stockwell, from the Greek word ferrum for iron, and as you point out, ptosis for falling off, and it didn't happen till 2012.
Yeah, because I don't remember seeing it until,
Yes,
but and we'd done a lot of work earlier exposing neurons to iron, and looked how it caused oxidation of the membrane lipids, and then you get these bits of the lipids that some of them can do bad things in the cell.
right. I think a lot of the work you did, like with 4-hydroxynonenal, and so, but the interesting story here is that around 1991, when I was still a resident, this group, Crapper McLaughlin and a group in Canada, had shown that intramuscular desferoxamine
Oh, yeah.
could, for 2 years, could essentially stop the progression of Alzheimer's disease. And so
So sorry to interrupt, Raj, but so at that point, I was at University of Kentucky, right? And
yeah,
Bill Marksberry was kind of my mentor from the standpoint of the, pathology and stuff. And they had this method. It's kind of like atomic absorption spectroscopy. Essentially, it's called laser activated something elemental analysis. But anyway, they'd have these sections of tissue from a person with Alzheimer's or control, and they'd put it in this llama device, and you can kind of visualize where the cell bodies of the neurons are, and they essentially burn that with a laser, and they get the spectrum, and depending on what wavelengths or whatever, you can tell. So they looked at aluminum, right? There's kind of this, not based on any science really, but this notion that aluminum pans were causing Alzheimer's disease, but one of the things they did find was iron
interesting.
in the neurofibrillary tangles. Yeah.
Interesting. So we, as I was reading the literature, and I wish I had access to that technique, but as I was reading the literature, I realized that we don't really understand how iron chelators are protecting neurons. So we decided to see what they did to glutamate, oxidative glutamate toxicity. And as expected, we got complete protection. And it was dependent on iron, because if you iron loaded the DFO, it didn't protect. And if you used other iron chelators, they protected. But what was really interesting is, as you probably remember, we used to measure cell death, we used LDH release,
Mhm.
which was, developed, I think, in 1987 by Dennis Choi in
Yeah,
his lab. And the idea was you would measure the amount released into the medium as a function of total LDH in the dish. And that,
just as an aside, I'm trying to, for people who aren't necessarily into the
oh,
science. So if someone has a heart attack,
right,
right? Well, they use some other things now, but uh, so whenever this LDH molecule, when a cell's alive and its membrane is attacked, it stays in the cell, and there shouldn't be any outside the cell.
that's right.
So if you have a heart attack and your heart cell, some of your heart cells are dying, there'll be an increase in LDH in the blood, and that's one way they, in addition to other things they use to diagnose heart attack.
Yeah. So, and I think that's so in essence, you're right. It's an enzyme that's released from the cell only when it's dying.
Yeah.
So you can actually measure the enzyme activity outside of the cell as a marker for whether cells are dying or. And the assumption of that method is that if you treat with a drug, that the relationship between the activity of the enzyme and the number of cells should stay constant. Well, what we found is that even though the cells were completely protected, and so in essence, no LDH was released, the amount of LDH activity, the amount of enzyme activity of this enzyme went up by 400%. And so I think for people who aren't familiar, and I'm glad you reoriented me because I forgot we're talking to a more general audience, lactate dehydrogenase is an enzyme that when your cells don't have enough oxygen, or if your mitochondria are not functioning, they help generate a molecule called NAD plus that allows you to generate enough energy by essentially revving the glycolysis engine up. And so we hypothesized, and there was a thought then that the majority of bad oxidants actually came from mitochondria. And
Mhm.
so the thought was, well, maybe what DFO, desferoxamine, the iron chelator, maybe what it's doing is it's somehow either making the cell believe it's hypoxic, or maybe it's inhibiting mitochondrial function and thereby reducing the load of oxidants from the mitochondria, but allowing them to generate enough ATP from glycolysis. And when we looked by immunoblot at all the glycolytic enzymes, remember there are a number of enzymes that convert glucose into pyruvate, so it can be used by the mitochondria. That pyruvate then goes into lactate by LDH. All of the glycolytic enzymes were increased. And then and we actually were able to show that the effect of the iron chelator was transcriptional. And we were stuck, and I was sitting in the I used to go to the Welch Library on Wednesday and
Yeah.
on Sundays. And I was always impressed because there's a very famous cancer biologist named Bert Vogelstein, who also I used to see there. And it was before PubMed. And what Bert used to do is he'd be thumbing through the journals, and then he'd be looking through almost every journal, and then he'd finish, and there'd be like this bonfire of journals on the table. I realized, I couldn't do that. I wasn't a member of the National Academy of Sciences, but I still look through all the journals, and I come to JBC and I find this article, and it says the transcriptional regulation of glycolytic enzymes by the transcription factor hypoxia-inducible factor 1. And in that paper, Greg Semenza showed that iron chelators are hypoxia mimics that upregulate all the glycolytic enzymes. And so I was very fortunate. I thought, oh, and this guy is at Hopkins. So I called Greg up and I said, Greg, I think we've just rediscovered what you've published. Can you help me? And to make a long story short, what we were finding was that iron chelators were stabilizing HIF1, upregulating all the glycolytic enzymes. And the evidence that, and that's really what got us interested in the whole idea. So for people who aren't familiar with HIF1, it's hypoxia-inducible factor 1. And it's a protein that is expressed in all the cells of our body. And as we sit here with normal, sufficient oxygen, there are a family of enzymes that are oxygen sensors called the HIF-prolyl hydroxylases. And those enzymes take oxygen, iron, and 2-oxyglutarate as cofactors, and they convert it to a tag on the HIF1 molecule that tags it for degradation. So in every cell of our body, HIF is being synthesized and degraded. But if oxygen falls below a critical tension, these enzymes, these oxygen sensors fail to function. HIF is now not tagged, it's stabilized. It dimerizes with its partner, which is constantly expressed, and then it moves into the nucleus, and it transcriptionally upregulates every gene that you would want to be upregulated if your body were hypoxic. It is the most elegant adaptive system. And I looked at this and I said, there was this, I said, this could be the mechanism by which iron chelators are so broadly neuroprotective in the nervous system, because there were 100 genes that were upregulated. They worked at a cellular level, a local level, and a systemic level. And it was a system that evolved over millions of years from metazoans. And I thought, this is fantastic. And so the critical experiment that we did at that time was, it turns out that cobalt chloride is also a hypoxia mimic. And cobalt chloride not only activated HIF, but it protected cells from ferroptosis. And it
That's
had
a little complicated because it also inhibits calcium channels.
Yeah, so
At least
it was consistent with the overall hypothesis.
Yeah.
You're actually exactly right. That was a complicating factor, but we did later experiments where we actually molecularly manipulated these enzymes and also pharmacologically manipulated them. But I think I think it was very formative for me, and then I think one of the issues, I just gave a talk on this. One of the issues we deal with right now is work we do in animal models. Young animals, usually genetically identical, not always. It often translates poorly to humans because we don't have the comorbidities, we don't have the genetic complexities, and so it really makes it difficult to believe that we can define single targets that are going to affect this broad array of pathologies that exist. And I think one of the exciting things about these transcriptional programs that are involved in stress adaptation is that they involve large numbers of genes that have the ability potentially to tackle that pathophysiologic heterogeneity. And that's where I would really like, I think we don't think that the iron we think they're still inhibiting an iron and 2-oxyglutarate dependent dioxygenase, because now we know there are many enzymes like this, but we don't think it's driving HIF expression. And but I think that I would be very eager at some point to hope to see this idea get tested in humans.
Yeah.
So
as you mentioned, these animal models, for example, stroke model, again, you have genetically pure animals and you occlude the exact same artery in the brain for the exact same amount of time. And, so you can pick out effects, statistically significant effects, if you do enough number of animals of some intervention. But then when you go to the humans,
Yeah,
There could be some effect, but you can't pick it out because there's so much, air.
I
Um,
totally agree. I totally agree. You can't, there's not enough of an effect. You need 1,000 patients in order to pick up the signal.
yeah.
And often we don't do studies with enough patients to be able to do that.
And as far as the stroke's a really good example where it's been known for a long time, and you talked about hypoxia. So obviously, when the blood supply is reduced to your brain, there's less oxygen, hypoxia. There's also less glucose, and but it's been known for a long time subject, if you occlude an artery, so induce hypoxia briefly,
Yes.
a little bit, and then you wait hours or the next day or next day, and then you come back and you do a full-blown stroke, there's some of the neurons survive that would have otherwise not survived had not you exposed to hypoxia. And I used to exercise a lot. I've got some issues now. Actually, I've got a channelopathy.
Ah, sorry to
Yeah.
hear that.
Yeah. So I got it's something called primary erythromalacia.
Huh.
It's a mutation in a voltage-dependent sodium channel that's expressed in nociceptive neurons.
Huh.
And so I have this hypersensitivity. But anyway,
And that only popped up later, or did you have that?
yeah, well, no, I've had pain issues. I don't know if when I came up there, that's been 20 years, I don't know how long ago, to
Yeah.
the Burke, right? I came up and we'll talk about this hopefully in a minute. At your rehabilitation facilities, you got these robots that
Yeah.
precisely. But anyway, I've had issues where like just sitting down on a hard surface, I get like really bad pain. So I sit for decades, I've sat on cushions.
Hmm.
Uh, but
This
anyway,
is this
so
is the
I'm
condition
so
that,
I can't exercise as much as I used to. To get back to the hypoxia, now you can buy, or you don't need to buy anything, but you can go on Amazon or whatever and buy little cuffs to go, say, above your biceps muscle and kind of partially shut the blood supply off. And then, if you do your resistance training, um, and this has actually been shown clinically,
yes,
you can kind of accentuate, you can build muscle with less actual, you know,
exercise.
exercise.
Wow, that's incredible.
Yeah.
So I that and is that thought to be is the mechanism of that
No,
known?
it's not known. So
Huh.
maybe you can look at.
Yeah.
Let,
Well,
let's
we
get to let's move on to how you are translating some of this uh, in animal models, and then kind of what you're envisioning for the clinic. One thing that we both worked on, and actually we did some when I was in Kentucky in the late 90s, is so we did a lot of work with caloric restriction, right?
yes.
Reducing calorie intake. So reducing carbs, reducing glucose intake, right? And maybe go into a ketogenic state. And so we wondered, and it turns out that we'd also worked on these stress proteins. There's heat shock proteins, but then there's another one in this, what you call ER, endoplasmic reticulum, these compartments that seem to be important sensors of cellular stress. One of them, it was initially called BIP,
Right.
but now most people refer to it as
Sure.
GRP78, glucose regulated protein 78. And
Yeah.
it was discovered by Amy, Amy something at UCLA. She's I think in the cancer field. But she discovered it because she exposed cultured cells to 2-deoxyglucose, and she found that expression of this gene encoding the GRP78, this important, it's called protein chaperone in the endoplasmic reticulum, it just shot up. And we'd done some work on GRP78 showing it was neuroprotective by manipulating it.
I know,
And
I'm familiar
then,
with that.
and then, so then we did some cell cultures with 2-deoxyglucose, pre-treating and protecting against glutamate or other insults. And then we did some in vivo studies. And then you you've done some interesting work with 2DG. I think this is something that could be used in the clinic, and for our viewers and listeners. So the 2-deoxyglucose, drug companies probably aren't interested in too much
Yes.
because I guess maybe if they had a use patent, like maybe you have a patent. We never actually filed a patent protection
Yeah,
on it.
I think we filed a provisional, but we haven't followed it up
Yeah.
because our commercialization committee said it's unlikely that it would have
You
uh
know, I did. There's another thing that another stress inducer besides, so 2-deoxyglucose kind of causes caloric restriction at the cellular level, if you want to look at it kind of a superficial level.
right.
But there's this molecule called 2,4-DNP, 2,4-dinitrophenol. And
Huh.
essentially it causes a lot of stress in the mitochondria.
Huh.
And, we found it was neuroprotective. And then there's this company called Mitocon that they actually have somehow through philanthropy and stuff, then they've got some FDA approval. They're doing some clinical trials of this toxin, but at low concentrations, it's very safe.
Well, I think as you know, we actually found that at remarkably low concentrations in animals that 2DG has these similar beneficial effects to what you had described. Although I think it, I think as you've pointed out in the past, it's clear that the metabolic switch that can happen to ketones may be important for a particular set of conditions. There also may be challenges in the long-term use of something that creates ketones. And I know I just saw a recent paper that suggests that chronic ketogenic diet can actually induce senescence in cells. But
And
so
chronic, we did, I don't think we ever published it, but Don Ingram, I don't know if you ever met him, but he was kind of a senior person, but he ended up in my lab we done some stuff with the 2DG. He supplemented the diet of rats with 2DG
huh,
and looked at lifespan.
huh.
So they were eating 2DG every day, and it shortened lifespan. And they did the pathology, and there were some problems with the heart. So I think, continuously taking this is not good, but maybe intermittently might be good.
Well, I think one of the things we're exploring is, and I think it's kind of ironic. It just shows that there are really no new discoveries because essentially what our focus is that low doses of 2DG. In fact, we're collaborating with a group at the University of Miami, Ted Lampidas, and they're using 2DG for cancer. And the concentrations they have to use because cancer cells love glucose,
Yeah.
and the concentrations that they have to use in humans are estimated to be 60-fold higher than what we used in our studies. And the idea is, our hypothesis is that, as you know, glucose can go either into generating ATP, or it goes into this process called N-link glycosylation, which is in the endoplasmic reticulum, which just makes proteins fold properly.
Yeah.
So if you inhibit the N-link glycosylation, the proteins don't fold, but they fold in a way that is not lethal to them. They unfold, and then they activate this ER stress response that ultimately drives genes that are involved in plasticity. And I think this concept is very much aligned with something you've written about, and that is that it makes total sense that there would be evolutionary pressure to drive genes involved in learning and memory when somebody was starving. Isn't that right?
Right.
So this provides essentially the detailed mechanism by which that occurs. Now, the question that we're trying to address now pharmacologically with Ted's help is there are analogs of 2DG. So glucose is a 6-carbon sugar. On the 2-carbon, 2DG has no hydroxyl group. What you can do is, instead of sticking a hydroxyl group, you can stick a fluoride. So it's F fluorodeoxyglucose. And this is used, I think people may be aware of it. It's used commonly in PET scans to try to look for cancers, for inflammation. And that's because the cancer cells and the inflammatory cells take up more glucose. And so you can actually see that on a PET scan. But it also turns, so the fluorodeoxyglucose has, so that the hydrogen on that carbon can be either up or down. So if it's down, it's FDG, but if it's up, it's what's called fluorodeoxymannose, which, so we always, when we think about sugars, we think about glucose, but there are other sugars like mannose that are critical for cell function, including N-link glycosylation. And the difference between glucose and mannose is the concentration of glucose is about 5 to 6 millimolar. The concentration of mannose is about 300 micromolar. So the concentration of a drug that might inhibit mannose's effects are much lower than glucose. So what we're doing is we actually have a grant that's being reviewed right now where we're proposing that fluorodeoxymannose would only affect ER stress and not affect glycolysis. And our prediction is that that drug is going to be much more effective as an intermittent treatment, maybe a real mimic of intermittent fasting. And it's not going to have the toxicities that you're going to see with other drugs.
So you can induce the adaptive stress response without compromising energy metabolism.
Exactly. That's exactly what we want to do. And I think that it's going to be a really, so we're proposing to test all 3 compounds, because maybe you want a little bit of both, but this is really a chemical strategy to be able to tell us, at least in an Alzheimer's model, which approach is better. And I think we have strong predictions, but I think it kind of gets to what you're saying. There may be lots of analogs of nutrients. I mean, N-acetylcysteine is an analog of a nutrient. There are lots of, nicotinamide riboside. There are lots of analogs of nutrients that are ripe for potential interventions in humans. And I think one of the things that we have to figure out is, do these just become proof of concepts that because the definitive studies are supported by the government, or is there a way to commercialize these so that a company can actually make money off of them, and then they can be distributed? But I think the third thing is their lack of expense potentially makes them deployable around the world, even in countries where the cost of the healthcare budgets are much lower. And I think we're really excited about this work. And it's, we essentially rediscovered what you already discovered. You already showed that 2DG upregulates GRP78. GRP78 is one of the ER stress proteins that's upregulated. So it's clear that we were looking at the same phenomenon. And the fact that we all are getting robust effects in animals suggests really I think it also suggests at how reproducible and the response is. And I think figuring out how to deploy it and what's the intermittency is key, because I think intermittency of stress responses is essential, and we can talk more about that whenever you like.
So essentially what you just described, and you really didn't mention the models, but you had a paper in Neuron last year.
Yes.
Where you showed these low concentrations of 2-deoxyglucose protect the brain against experimental stroke, and then used a complicated genetic mouse model of Alzheimer's where the animals get amyloid pathology and they also get tangle pathology.
No,
No,
not
it's just a lot
just
of mutations that increase the amyloid, I guess.
that's right. And
Um,
a lot of inflammation, very aggressive Alzheimer's model,
and cognitive impairment, which you
cognitive
measured. And
impairment.
you showed that this 2-deoxyglucose not only protected the neurons when you look at the brain, or the amyloid, but also improved the functional outcome in motor function in the stroke model and learning and memory and the Alzheimer's. And I think this is really exciting, and it's unfortunate way that, as you kind of alluded to, the way the system's set up. Um, big clinical trials are almost always supported by drug company money,
Yes.
right? And they aren't going to do clinical trials with something that they don't have a patent on or can't make money. But on the other hand, there are many things. When I was at Kentucky, we'd done some work. Again, this is another example of something inducing kind of mitochondrial stress response. There's a drug called diazoxide.
Hmm.
It was actually used to treat hypertension a long time ago. It's very potent. It opens potassium channels and causes, well, relaxation of the membrane, decreased excitability of the cell.
Hmm.
But there's also these potassium channels in the mitochondrial membrane, and it'll do. And we found that was very neuroprotective. And there was I don't know if you came across this neurologist, Wes Ashford was at Kentucky, and he was clinician seeing Alzheimer's patients. And this is an off-patent drug. It was FDA approved for hypertension. So he, without any protocol in place or anything, he prescribed it to some of his Alzheimer's patients. And this is anecdotal, but he claims it was having beneficial effects. We went to the company that initially made the drug and got the patent for hypertension long 40 years ago now, 50 years ago, whatever. It was one of the first drugs for hypertension, and they didn't have an interest in it. This kind of, so it may never get tested.
Yeah. Well, I think that's, I think the government has set up a lot of systems to be able to potentially test drugs where, I mean, Gary Gibson here at
For a long time. Yeah.
yeah. So Gary has a 40-center, $45 million NIH study using a drug called benfotiamine, which is also, not patentable.
Yeah.
But the NIH supported this effort, and I think if benfotiamine were to be beneficial, then pharma would show interest.
Yeah.
So I think the NIH has done a great job of creating structures that allow us to take these
Okay, good.
agents that are off patent and potentially test them. And I think, it really requires the energy and commitment, though, of, getting the IND going forward.
One thing that happened when we were doing all this work on intermittent fasting, and I was at NIH, and of course I know the administration there all the way up to the director. And so as the data started coming out and stuff, they finally got interested. And then finally, I think 4 years ago, they put out an RFA for clinical trials of intermittent fasting in humans. A lot of money for, cancers, neurological disorders. And there's now, if you go on clinicaltrials.gov, there's over 200 trials of intermittent fasting.
Fantastic.
So have they put out, so there needs to be kind of there isn't really that kind of a stimulus, is there? Or is
Well,
it?
I think that I think the things I would say that probably not that great a stimulus, but there's an urgent need. There are infrastructures for actually doing trials. And
Yeah.
I think what catalyzed the movement of Gary's studies with benfotiamine forward is they did a 100-patient pilot trial here at Berk where the primary endpoint failed, but all the secondary endpoints were improving. Now, that's a very highly underpowered study. Was only 70 patients. But I think that kind of smoke in humans goes a long way,
Yeah.
isn't that
Yeah.
right? And
Yeah.
so to the extent that the institutes support these pilot trials that give confidence about safety
Yeah.
and some evidence of efficacy, then I think they can grow into larger phase 2 and phase 3 studies. And I would say, we probably have 5 examples now in the lab of drugs that target these stress sensors and how they can be potentially beneficial. And so I think, and we didn't in very like with 2DG, we never started out thinking we would end up with an adaptive stress response, but that's where we ended up. And so it's and I think that's where I find a lot of resonance with your work. It makes a lot of sense that you want drugs that are going to drive a homeostatic response, that the goal is to restore homeostasis, and the body knows how to balance all these different priorities. Isn't that right? It's sort of figured that out over millions of years of evolution. And now we have just an army of small molecules that potentially can affect these responses. I think the keys, though, are, as you point out, that they have to be given intermittently. And the question is, how intermittently?
Yeah.
Is it once a week enough? Is twice a week enough? And maybe more of the studies need to be focused on figuring that out.
Yeah.
I think that the persistence, if they're persistent, so we often look for these responses in autopsy brains, isn't that right? Or we look for evidence of, and the persistence of these responses is a bad sign, because if you have persistence of these stress responses, the cell says, I'm not compensating.
We have to have time to recover.
Yes, you need a recovery phase. And so I really think that there is a logic that's coming together.
Yeah.
It just has to be that we have to try to codify it. Isn't that right?
Yeah.
To really augment treatment.
Do you want to talk a little bit? Do you still have a few more minutes?
I do.
Okay. So there's 2 other areas you've worked on with regards to translating, basic work to the clinic. One has to do with, so we're talking about stress responses, this antiviral stress response. What the heck's that got to do with protecting neurons?
Good question. So, do you want the long or the short version?
Well, this is the area go ahead.
Okay. So I'll tell you, the way we got interested in this is that, and it really dovetails with work other people have done, is that when I was still at Harvard, we did a screen. We were very interested in FDA approved drugs that would activate hypoxic adaptation.
Mm-hmm.
So we stably expressed a HIF reporter in a hippocampal neuroblast, and we screened drugs to find which one could drive the luciferase reporter, which it's just the luciferase from fireflies. So you measure light.
Yeah.
And I had a visiting scientist from Taiwan and a neurologist, and I said, this will be a good project for you. And what he found is that the number 1 hit in that drug screen was a drug called Tillerone. And Tillerone turns out to be the first antiviral immunomodulatory drug. It was developed in 1960. And we showed that if you treat with Tillerone 12 hours before inducing an ischemic stroke, you reduce the stroke size by 90%. And initially we thought, oh, well, this is working through the HIF pathway, but I had another visiting scientist come to the lab and I said, it doesn't look like it's activating HIF. Maybe you should check out whether it's activating the antiviral interferon response. So it was initially used to treat the flu, and it lost FDA approval because it had some side effects. And lo and behold, that drug was massively inducing interferon genes in the brain. And there have been two observations by other scientists that really catalyze this. Mary Stenzel-Poor in Oregon she was an immunologist, and she took hypoxic preconditioning. She took LPS, which is a part of what makes bacteria bad for you, and she took what's called poly-DIDC, which is kind of an RNA mimic. And when she used all of these agents, they could precondition the brain against stroke. And the commonality of all of these three agents was 13 interferon genes. And they were many of the same genes that we found upregulated by Tillerone. Now, when we were looking around to find out what Tillerone actually does, we found that its action is that it inserts itself into DNA. And in 2013, a guy named James Chen, who was working at UT Southwestern, discovered this sensor for, so DNA is in our nucleus and it's in our mitochondria. It's not supposed to be in our cytoplasm.
Right.
But if it's in the cytoplasm, that is a danger signal that says maybe I'm infected by a virus, or maybe my mitochondria and my nucleus are damaged. And what happens is the double-stranded DNA, if it's greater than 45 base pairs, it binds to a protein called cGAS. And cGAS generates a second messenger, and it's a dinucleotide second messenger called cyclic GAMP. And then that activates a series of downstream proteins, I won't go through all of them, that ultimately turn on all the interferon genes. And so we initially thought, oh, well, maybe Tillerone's just stabilizing DNA and activating cGAS. That turned out not to be the case. But in doing those experiments, we were led to the fact that there are now small molecule activators of the downstream target of cyclic GAMP. It's called STING, stimulator of interferon genes. And there are drugs that activate this pathway. And if you give these drugs to an animal 12 hours before stroke, it completely, there's practically no stroke. And if you do it in an animal that doesn't have STING, it's not protective. So now we feel like we've mapped the pathway.
Yeah.
That and this stress response, and there is an exciting paper by Tony Whisk-Coray, who's done a lot of the parabiosis experiments. He showed that ganciclovir, which is thought to be an antiviral drug, it actually can act as a STING agonist.
Huh.
So there may be drugs that we can actually move into humans very quickly that can activate this pathway that. And you might say, well, why would the interferon response be so protective against stroke? Well, if you think about it, if any cell in our body thinks it's infected, the interferons are there to essentially shut down macromolecular synthesis. It turns off translation so that the virus can't replicate, and essentially shuts down metabolism. So the ATP demand goes way down, and the ATP, so it balances the ATP supply with the demand. Now, I will tell you, what's really exciting is that there's a flip side to this response, which you're probably aware of, Mark, and that is a beautiful paper published in Nature about a year ago by Andrea Blosser, which showed that in aged mice, if you start at 20 months, maybe the equivalent of what would be 65 for us, okay? And you give a sting antagonist to the time the animal is 26 months, you completely block the degeneration in the hippocampus and the memory decline that happens with aging. And so it appears that this response, as we age, for reasons that are still obscure, in the paper they speculate that during aging, your mitochondria become leaky and the DNA starts getting released into the cytoplasm that then activates cGAS, which activates cyclic GMP, sting, and then upregulates all these interferon genes that then downregulate memory. And what we have a paper that we're putting together that suggests that this, so they hypothesize that it takes 6 months and you need hippocampal degeneration. What we can show is that a single dose of sting agonist is sufficient to reduce LTP, long-term potentiation, a form of synaptic plasticity in the brain, and reduce learning and memory. So you don't, and this may be why, you know how the morning after you have a cold, why when you have so much trouble getting your head off the pillow and you feel this kind of brain fog, we think it's because of this stress sensing pathway. But and so blocking it before stroke is a good thing. What we're finding is that if it's persistent, it may contribute to cognitive aging. And also it may be activated after stroke to limit stroke recovery.
And has someone been looking at this in relation to long COVID?
Yeah, so the data is people have been looking at it, and I don't think that and the data would be maybe consistent with it, although there are other models. I think the problem is that you have to be able to assess what's going on with the cGAS sting pathway in a living human, and I think that's where PET ligands hopefully can be developed that actually allow one to be able to look at this pathway. But it turns out that there are FDA approved drugs. So one of the hypotheses is that cGAS and sting get activated in a cell type in the brain called microglia. They release these chemokines, CCL3, CCL4, and CCL5, and they bind to a receptor called CCR5. And that turns out to be the HIV receptor. And there's a drug for that blocks that called Maraviroc. And Alcino Silva at UCLA and Tom Carmichael there have used Maraviroc in animal models with great success. We have, following on their work, and Maraviroc could be used in humans immediately. So I think a careful trial of Maraviroc in long COVID would be important. My concern is I think long COVID is probably many things.
Yeah.
Like if you have pulmonary dysfunction, maybe there's some hypoxia. If you have cardiac dysfunction, maybe there's some hypoperfusion. If you have, so if you're depressed, for other reasons, then I think that can contribute. So I think I did a podcast on this. I'll be happy to send it to you, where I tried to create an algorithm for the evaluation somebody might go through to ultimately ferret out what might be the culprits in causing their brain fog.
Yeah. And one more example. So you had a paper in Cell in 2019 with showing that selenium can be very neuroprotective. Can you talk about, so we talked a lot about iron ferroptosis. So this has an interesting thing from an evolutionary perspective, because
Yeah.
it's thought that the first life was in these deep ocean vents, volcanic vents.
Yes.
They had very, very high concentrations of iron, hydrogen sulfide gas, and metals in general, I think. And so
Yeah.
iron, selenium, arsenic, people have heard know it's toxic. But iron and selenium are very interesting because they can be very toxic, but we've evolved so many interesting ways, not only to protect cells against the toxicity, but to actually use those metals to their advantage.
Yes. Right. Yeah. So I think you're right. I mean, as maybe the metals were there before oxygen started to evolve. So as oxygen was utilized, there had to be mechanisms that were developed that counteracted
Yes.
the toxicity, that leveraged the high reactivity of oxygen with metals in the form of ATP and mitochondria, but also that evolved antioxidant enzymes that could withstand the stress that was imposed simply by using oxygen for energy. Is that right? So as you probably know, these selenium, when it's incorporated, sorry about this, from the hospital, the when selenium's incorporated as selenocysteine, it has a lot of advantages over cysteine in that its properties are such that it can be oxidized. That is, it can lose an electron, and it can gain an electron back very rapidly. Whereas with cysteine, it can be modified irreversibly by oxidants. So our bodies have evolved selenoproteins that resist this oxidative damage. And there are 25 selenoproteins. But the redox activity of selenium also potentially gives it a very narrow therapeutic range. Now, what's fascinating about ferroptosis is its most essential antidote is an enzyme called glutathione peroxidase 4, which is a selenoprotein. Okay. And so in order to, and the thought is that, so the simple idea was that when you add selenium to cells, that you would optimize glutathione peroxidase 4 activity and protect against ferroptosis. And that's what we found. What surprised us and what was unusual, and again, it kind of gets back to the fact that we keep being dragged back to the nucleus, is that we found that if you give selenium to a neuron, it upregulates GPX4, but it requires new transcription. And it suggested to us that, and what we found was that not only was selenium, and this was pharmacological levels of selenium, so the neurons have totally normal nutritional levels of selenium. If you add supranutritional levels, you actually drive the expression of all the selenoproteins. And it kind of makes sense because here you have a metal that's kind of toxic.
Yeah.
The cell is saying, I've got too much. What am I going to do? I'm going to upregulate all those proteins that would incorporate that selenium and use it. Isn't that right?
Yeah.
So we did an experiment where we injected selenium directly into the ventricle, one dose, and we induced a brain hemorrhage, and it protected. But what we found is that there's a parabolic dose response. And I think this is something you highlighted, that even though there's a protective concentration, you reach a peak and then it starts to become toxic. And this is a real problem therapeutically because you never know. So we had this idea that the chaperone protein around the body is a protein called selenoprotein P. And it's a unique protein in that it's C-terminal end. There's an N-terminal and a C-terminal end of the protein. The C-terminal end of the protein has lots of selenocysteines. And so the simple idea was to take those amino acids, 6 amino acids from the C-terminal end with 2 selenocysteines, and link it to a TAT protein, which is a part of the HIV TAT protein it's 11 amino acids that when you link it to this peptide, it now can get into the brain on its own. And so we did that, and we first did it in vitro, and lo and behold, what we found was that the parabolic dose response disappeared. It became sigmoidal. If you incorporate selenium into selenocysteine, you now have this amazing dose response. And the advantage of just taking selenocysteine is now you can imagine using a targeting peptide to target it wherever you want in the brain. Isn't that right? And so we were able to show that it not only blocked ferroptosis in vitro, but it blocked hemorrhagic stroke and ischemic stroke. And again, I think I'm very excited to kind of take peptides like TAT NR2BC that Mike Timiansky developed for stroke, or other peptides that have already been shown to have some effect, and simply stick the 6 amino acids onto the end.
Yeah.
Because now you get this amazing upregulation of 384 genes we call the selenome that block ferroptosis, ER stress-induced apoptosis, excitotoxicity. And I think it is, again, another just beautiful example of how these sort of endogenous programs are ripe for treatment in humans.
Yeah.
And I think that, we don't understand what the sensor is there. I'm very interested in trying to figure that out. But I think it is an exciting line of investigation, which kind of fits well with the theme that's been running through our research throughout my career.
Yeah. Yeah. So this idea of, challenge or stress recovery cycles, I'm using the word challenge a lot any more than stress, because stress sometimes has a bad connotation.
Right.
But, exercise is a challenge, fasting is a challenge.
Yeah,
At the cellular level, the cells do perceive it as a stressful, potentially dangerous situation if it continues for too long.
yes.
And
I like the
so
word
there's
challenge.
for translation, there's this, this is kind of a new idea.
Yes.
If you look at the big picture, and the idea of intermittent dosing is very foreign to drug companies and so on. In fact, usually they develop a drug so that they want it to kind of get steady, called steady state levels. But and those drugs are almost always blocking blocking one particular pathway or has one particular target. But in this case, the idea is to activate a sequence of gene expression that's evolutionarily conserved to deal with stress.
Yes.
And then you're, in theory, protecting the cell in multiple ways against many different kinds of
Absolutely.
stress.
I think that's it. And I like the word challenge because I think that it, sometimes I think people don't realize that if you go and you exercise, but it may be better to do short periods of exercise where you really push yourself. Isn't that right? Than it is to kind of, but I think the issue is that maybe challenging the boundaries of homeostasis is a way of bringing the system back into a homeostatic
And
balance.
if you don't challenge the system, it becomes complacent.
That's right.
And not able to respond adaptively. So
and I think that would be the downside of taking antioxidants continuously. Isn't that right?
right, and there's been, I mean, trials failed,
Yes.
and the guy Ristow, right? You know, he's worked in, I think he's in
Yes.
Germany, showed with exercise, if you load up people with vitamin E and have them exercise, they actually, their performance goes down.
Ah, interesting.
Yeah, and so these free radicals have important signaling functions, right? And if you stop
Absolutely.
them all up, it's not good either.
You know, it's so ironic because, you know, there's this practice, which a lot of people are skeptical about, called homeopathy, where you take small amounts of
Well,
substances
yeah,
to,
yeah.
but I think what we're talking about is kind of a molecular form of that, isn't that right? Where you're really trying to
Well,
sort
we'll talk, there's another word that maybe some people get confused, hormesis.
or
And
mesos,
this is just the idea of a biphasic dose response, you know,
right?
that low to moderate doses of something can do a good thing, but if you get too much, I mean, this is old wisdom, everything in moderation, right?
It is the empiric distillate of many, many years of experience, isn't that right? And all we're doing is kind of putting a molecular face behind it
Yeah,
now. And that's where I think, I think it's a very optimistic view, because it really suggests that people can be doing things in their daily lives
that's right.
to challenge themselves.
Yeah,
And that is, as long as they're giving themselves enough time to rest
right.
in between, that
So,
that can
so
really,
focus on, so listeners and viewers, focus on what Raj and I are talking about. That's a challenge to your neurons.
right.
And then get a good night's sleep.
That's right.
That's
Well,
the reason.
well, you know, and interestingly, I mean, I think one of the things we speculate on in the neuron paper is the idea that, you know, if you stop eating at 6 o'clock, if you don't eat after 6 o'clock, by the time you sleep, you may reach a level of fasting to actually activate the stress response. Isn't that right? Where you're going to decrease your end-link glycosylation. So
Maybe
while
by,
you're
by
sleeping,
morning anyway.
yeah, by morning. So maybe sleep, actually, some of its benefits involve the fact that you can actually activate these adaptive.
Who's the up at University of Pennsylvania, the sleep group up there? There's a woman and an older guy who they've looked at ER stress response and sleep or something
I
like that.
think we found some papers where people have shown ER stress goes up,
Yeah,
autophagy goes
yeah,
up, and it is interesting that, um, you know, people talk about the fact that it's, you know, continuous sleep rather than fragmented sleep, and maybe there's something about the fragmentation that actually disrupts
yeah.
these stress responses. So it may be that you can get, but again, I would say that one of the things we're hoping to do is, if we can start to get some blood biomarkers
Hmm,
of what's a when is BDNF actually increased in the brain,
yeah.
then I think each of us can figure out what's our right dose. Isn't that right? Of how long do
Are
you need?
you started or getting interested in or collaborating with someone on extracellular vesicles? And
I haven't, but I would love to do that. I think what we're planning to do is to, initially, the idea is to simply do single nuclear RNA sequencing on cells of the blood in animals, and
okay,
correlate that with what's going on in the brain.
yeah.
If ER stress is occurring in the blood at the same time it's occurring in the brain, then that becomes
You know, there's a neurologist who was under me when I was NIA, Dimitrios Kapagiannis, and he's developed this method where he uses an antibody against the neuronal protein, membrane protein, and he isolates these extracellular vesicles from blood and then uses the antibody to pull down the vesicles that are probably coming from neurons.
that would be
And
fantastic.
and he's even shown that like with all mild cognitive impairment and Alzheimer's disease, so these vesicles, they have tau, the microtubule associated protein, and it's hyperphosphorylated. So he's kind of using this as biomarkers for what's going on in the brain. He even showed that in people with AD and diabetes, that these vesicles have the insulin receptor substrate protein 1. It's anyway, he can get indication of the relative insulin sensitivity
Of the brain.
of the brain by probing
So
these.
where is he? I'd love
He's at
to.
NIA.
Oh,
Yeah.
okay. Maybe I'll get in touch with him
I,
because
I was I
I
was his, I was his boss.
interesting. Well, the other thing I think that we haven't talked about, but I'm curious about is to what extent does 2DG interface with glucose sensing neurons that maybe are responsive to like GLP-1 agonists and things like that. I think
Oh, yeah.
so that it seems like there's a a vast array of things that we could explore, which we probably won't do ourselves, but we'd love to do collaboratively.
Okay, Raj, I appreciate you taking
This is terrific. This
time, and
is fun.
it's good to see you again. You're looking great, and
Thank you.
things are going well up there. And say hi to Gary, and
I will do
yeah,
that. It was great to talk to you. Thanks, Mark. Be well.
you too. Bye.